Click for IB Biology, Photosynthesis. In this video we'll be looking at the photosynthetic pigments, their arrangements on the thylakoids into photosystems, and the existence of what are known as reaction centers. We will take a quick look at accessory pigments and the processes of how to identify them and indeed how to image them. There's a PDF of this presentation in the description below, along with a quiz that you can use to test your understandings of the theory work covered here. So if you're lucky and you live in a temperate region of the world, this kind of site shown in this image here will be familiar to you. The temperate forests of the world in the autumn period, or the fall as some would call it, show these wonderful range of colors, of light greens to dark greens to reds to browns, which are a very, very beautiful sight to be seen. But for a biologist, there's a deeper meaning to this beauty, and that's what we're going to take a look at in this piece of theory work. So it turns out that on the thylakoid membranes of a chloroplast, we can see two different regions. In this micrograph, stained darkly are regions like this, which are called grana, and then they are connected by long membranes between other grana by the intergranal membranes here. So that's here. The pigments associated with the collection and harvesting of light are arranged into what we call photosystems. So the photosystem is the combination certainly of a chlorophyll, but with some additional accessory pigments. And they combine together to give what we call a photosystem. Now there are two photosystems, and typically photosystem I is on the intergranal membranes, so this region here, whereas photosystem II are organized onto the granule stacks, typically the darker regions seen here. So photosystem II on the grana, and photosystem I onto the connecting intergranal thylakoid membranes. So let's take a look at what photosystem is made up of. At the heart of the photosystem, as shown in this diagram here, is the chlorophyll molecule, usually chlorophyll A. And this is surrounded, physically surrounded, by additional pigment molecules. And you can see here that I've named two sets, which are chlorophyll B and the carotenoids. And the coloration that I'm showing is not incidental. The carotenoids are indeed yellow colors, and chlorophyll B is a slightly different green color. So physically, they are organized around the chlorophyll A, and the chlorophyll A is the reaction center that we understand that when light energy is captured by the photosystem, an electron will be released into the cell chemistry from the reaction center called chlorophyll A. So there's two ideas associated with these additional pigments surrounding our photosystem here. So the pigments which are surrounding it are understood to, first of all, protect the reaction center from high intensity damage of light. So it's possible that chlorophyll A could be damaged by very intense light, but this doesn't happen. So if we think about it like this, here's the chlorophyll A, here's chlorophyll B, and perhaps a carotenoid here, and very intense light is absorbed by the carotenoid. The energy is then stepped down with the release of an electron, and energy is then dissipated as it moves from one pigment to another, and by the time it's passed to the chlorophyll A, the electron is now at an energy stage suitable that it will not cause damage to the chlorophyll A. So it's protecting the chlorophyll. The accessory pigments protect the central reaction center from high intensity light. Another way of thinking about this, and that's why this image is shown, is that therefore at the same time this means that the reaction center chlorophyll can benefit from a broader range of light intensities that can all be channeled and stepped down in their energy level to something appropriate for the reaction center. So we can collect light from a broader range of intensities. You may have the opportunity in your laboratories to perform forms of chromatography. This one here I'm calling TLC, is thin layer chromatography, performed on a plastic strip with a chalky-like layer here instead of paper, and we concentrate our chlorophylls at this region here, and then they are extracted by a solvent that soaks up along the TLC plate and ends perhaps up here, and that would be called the solvent front, which is this here. And so we have a distance from here to here, which would be called the solvent front. And then there's a distance traveled by a particular pigment. Perhaps we'll select this one, and we can measure that distance as well, giving us the value to put here. Divide the two gives us the RF value, and from that RF value we can identify what the particular pigments present are. Although I should mention that the distance traveled and the particular RF values depend upon the solvents which are used in this technique. So in doing so, we can identify, as you can see in this image, some of the chlorophyll A's and B's, a number of the carotenes, and perhaps even a xanthophyll in this mixture, making up the photosystems. In the 21st century, microscopy has seen significant improvement to what is called crypto-electron micrographs, in which the images that you're looking at on the right-hand side are not measured in microns, but rather in nanometers, which therefore is 1,000th of a micron. And because of this, what we are actually seeing are molecular images. So the arrangement of the accessory molecules and the chlorophylls are actually now being imaged, not just the organelle. The link to our autumnal season that students may be familiar with is that in late autumn, the central chlorophyll molecule, with the falling light intensity of the autumn, the leaf and the plant no longer maintain their chlorophyll, and the dominant green color of this chlorophyll begins to fall away, and what's left behind are the colors of the accessory pigments, and those accessory pigment colors of the carotenes and the xanthophylls, those are the autumnal colors that we see at that particular time of year. Appearing in the video, in the window now, are the links to the next videos looking at absorption action spectra, a link to the channel that you can subscribe, and to the playlist for you to look at the variety of videos which are available to you on this topic of photosynthesis.